What a good day, Miss Moira. These are lab analysis techniques. First, formulation of SLN containing 2-nonanon and 2-tridecanon. The study synthesizes SLN containing 2-nonanon and 2-tridecanon using high-share homogenization and ultrasonication methods. The lipid phase is dissolved in hexane and the 2-nonanon or 2-tridecanon is added. The aqueous phase is dissolved in distilled water, homogenized and sonicated. The resulting solution is cooled and stored at 4 degrees Celsius until use. The SLNs containing 2Ketones are stored at 4 degrees Celsius.
Characterization of solid lipid nanoparticles. The morphology and particle size of SLNs were determined using STEM and TLS, while their size, polydispersity, index, and surface charge were characterized using DLS. TGA and FTIR spectra were obtained using Agilent Technologies carry 630 FTIR spectrophometers and with the transmittance between 3600 and 600 cm.
Quantification of 2-nanonon and 2-tridecanon released from SLN by HSGC-FID. The bioactivity compound released from SLNs contains 2-nonanon and 2-tridecanon was measured using HSGC-FID. Release curves were performed for 72 hours and triplicate aliquots for formulation were measured and diluted in hexane. Samples were heated and injected into GC-FID for quantification. The analysis were performed using a BPX5 capillary column and external calibration curves with commercial standards. The operating conditions were 250 degrees Celsius, carrier gas, and oven temperature.
Plant experiment and growth condition. The study evaluated the growth modulation of 2-nonanon and 2-tridecanon released from SLNs in L. sativa seeds. The seeds were sterilized, placed on moras seeds and scoop basal medium, and experimented in 2 compartment petri dishes. 3 to 4 day old seedling were placed on MSE and paper disc impregnated with SLN containing 2-nonanon or tridecanon. The experiment was conducted in a refrigerated incubator with lighting and a 16 to 8 day night photo period.
Photosynthetic pigments. The study measured and chlorophyll content of LA sativa left and sample and analyzing their absorbance at different wavelengths and using genus 2 SUV VIS spectrophotometer.
Psychological parameters. Electrolyte leakage was measured in a solution of deionized water containing 10 leaves with initial electrical conductance determined using a conductivity meter. Pre-Pulline concentration was determined using fresh tissue diluted in ethanol water solution. Samples and Pulline calibration curve were measured at 520 nanometer using a spectrophotometer. Total protein concentration was determined using the Bradford method, with samples incubated at 25 degrees Celsius for 10 minutes and absorbance read at 595 nanometer.
Determination of total phenols. The following CO-calcium method was used to determine total phenol contents by adding leaf extract to mixed reaction. Samples were incubated for 30 minutes. Absorbance was measured. Garlic acid was used for calibration.
Identification of picric acid. The study used HPLC-DAD to analyze samples using an SPD M20A UV VIS spectrophotometer. The Agilent 1100 series system was used for identification assessment. Chromatographic separation was performed using a a chromacell plastic shell, 2.5 C18 columns, and a C18 pre-column. Samples was injected with HPLC grade water. Acetyl-chromatic formic acid and water acidic formic acid was mobile phase identifies assessment were performed in an HPLC DAD QTOF MSMS compact.
Antioxidant capacity. Leaf samples were mashed in liquid nitrogen dissolved in extraction solvent, syndicated, and centrifuge. The first observance was measured using DPPH reticle, followed by cuprock and TAP measurements. Cuprock was measured using a mix of CuCl2, neocorpine, and ammonium acetate buffer. TAP was performed using ABPS 7.5 millimolar reagent with the first observant at 734 nanometer. The second observance reading was performed using 5 microliter of sample of Trolox standard. Results were expressed as Trolox equivalent.
Antioxidant enzyme activities. The study involved immersing L. sativa seedling leaves in liquid nitrogen and adding a mixed solution. The extract works, centrifuge, and the supernatant was used to determine antioxidant enzyme activities. The results showed APX, C80, POX, and SOD activity. APX activity was measured by adding 50 microliters of LAB extract to a solution of 0.1 millimolar EDTA, 0.5 millimolar ascorbic acid, 50 millimolar potassium phosphate buffer, and 13 microliter of hydrogen peroxide. POX activity was measured by adding 20 microliter of LAB extract to a solution of 0.1 millimolar EDTA, 13 millimolar methionine, 2 micrometers riboflavin, 75 micrometers chlororo-D-nitroblue tetrazolium, and 50 millimolar potassium phosphate buffer.
Statistical analysis. Experimental data were analyzed by statistic V10 Talahase FL USA to perform analysis of variance ANOVA difference among means of treatments were separated using the 2K test at 0.05 of significance.